Hello viewers, my name is Damilare. In this lesson, we're looking at the topic genetics. Genetics. From the word genetics, the first word that should come to your mind is the word gene, meaning that in genetics we'll talk about gene, but we are coming back to that. What is the definition of genetics? Genetics is the study of heredity. There is a word from this heredity, H-E-R. This use it to remember inherit, meaning in heredity we talk about what we inherit from our parents that made us resemble them. And we also look at variation. There is a word we can get from variation, that's vary, talking about what makes us different. That means when you look at twins, no matter how identical they are, you still see some differences in them. So meaning that when we are studying all these things, we are studying genetics, studying what we inherit from our parents, studying that makes us resemble them, and studying what makes one different from one's parents. That is variation. So the genetics is the study of heredity and variation. Now let's focus on variation. Variation is the difference which exists in individuals of the same species at corresponding stage in their life cycle. So we have said it earlier that variation means the differences you see between two persons that are of the same species. And I've also talked about heredity being inheritance, what one inherits from one's parents that makes one resemble one's parents, and that has to do with transfer. You inherit something by transfer of traits. Another name for trait is character. Like maybe your parent is tall, you are also tall. They are fat, you are also fat. That's a trait. So from one generation to another, that means that thing, that trait you inherited from your parents, you also transfer it to your own children. Now there are some common terms we use in genetics. Number one is gene. When you hear gene, what is gene? Gene is a physical unit of inheritance transmitted from one generation to another and responsible for controlling the development of characters in the new individual. Keyword units. When we say unit, that's parts. It means part of what we inherit is called gene. That means once gene enters your body, it begins to produce some characters. So meaning that is the unit, the foundation of what we inherit, the major thing we inherit from our parents. So that is gene. So it's a unit of inheritance, and gene is always being transmitted. That means it moves from one person to another, so it's being transmitted, and that's from generation to generation. And gene again determines your character. So 3 points from gene is the units. That means the beginning, the foundation, the parts of what you inherited, and it is always being transmitted. It moves from parent to children, and finally it always determine character, or it comes along with characters of a human. Now another word is chromosome. What is chromosome? Chromosome, they are strands of genetic materials. When you hear strand, something that looks like hair, that's what we call strand. Like you can say strands of hair, so that means it looks like hair. That's chromosome. As you can see what I drew here, something that looks like this X is called chromosome. So it's a strand of genetic material. That means this strand of chromosome, it has some genetic materials in it. It has what we call gene in it, and that gene is actually in the DNA, because chromosome still contains what we call DNA, and this DNA is now having gene in it. So the structure of DNA is the one we have here. So DNA is always a double helix strand. Double helix, like a strand curving to the right and to the left, so they are now interconnected. That's a double helix strand of DNA. So DNA now has certain other things. Even you can get it from the D. It has deoxyribose sugar inside DNA. There is the nitrogenous base. The nitrogenous bases inside this DNA. What are the nitrogenous bases? They are the adenine, thymine, guanine, cytosine. And take note, adenine and thymine, they are pairs, like they substitute themselves. Then guanine and cytosine, they are also pair. That's gene pair. So that is another part of this DNA. Then A, this A, you can just take it as P, maybe like nap. DNAP. So P means protein, meaning there is also protein in this particular DNA. Apart from protein, there is phosphate. It has a phosphate group. So the protein, is just like a polymer. So DNA can be in form of a polymer. It's polymerized. So they used to call it polymerase. So meaning that it forms a polymer because the DNA is always being reformed. Another one can be produced again. Another one can be produced again. And how will it be produced? There will be a combination of some codes. So those codes are the gene, meaning that from this middle here down to this place, there is a gene there because there is a code of different codes that form a gene. The same thing at the top and under it. So that is your DNA. So since I said genes, they are inside this DNA, and the DNA itself is also inside chromosome, and that's why indirectly we can still say that there is gene in chromosome, and that chromosome is always inside the nucleus of a cell, and cell is found in the living thing. So chromosome is a strands of genetic materials. It become obvious during cell division. It means when during cell division, that's when there is reproduction or growth, you see how chromosome begin to manifest, especially through the processes we have, all this interphase, telophase, prophase, metaphase, anaphase. So that's when you see chromosome manifesting during cell division. They are found in the nucleus, like I said, when they carry genes. They consist of deoxyribonucleic acid, DNA, as I've said earlier, and protein. So the name of that protein is histone. Histone is the protein that we find in the DNA, which is also in the chromosome. So they are always being packaged. So they are being packaged. They can also give shape, that protein. So they are packaged inside the DNA, as you can see it here, and they also give shape to that DNA. Now, another thing about chromosome is that we have 23 pairs of chromosome. For every human being, there is 23 pairs of chromosome. One pair is called autosome. That autosome is a sex chromosome. It's one that will determine if someone will be a boy or a girl. So that's the sex chromosome. But the remaining 22, they are the autosome. So this one is 22 autosome, which is the one that will determine other gene, other characters. Maybe one will be tall, one will be fat, one will have this kind of blood group. All those ones are the autosome, but the only sex chromosome is one that will determine maybe one is a boy or girl. So total is 23 pairs, and 23 pairs is the same thing as 46. It means every human being has 46 chromosomes, or you say 23 pairs. One is the sex chromosome, one pair, then 22 pairs is the autosome. So that's under chromosome. Then another thing is that for the sex chromosome, if one is a boy, that would be XY. That's it, because it's always pairs 2 for that sex chromosome of a boy. Then again is X. So, and these are the two things that will now determine. It means if this is a man, XY, and if this is a woman, XX. So it means the man from the Y will determine if a boy or a girl will be given birth to, because a woman will always produce X, while a man may produce X or Y. So if a man release Y and a woman release egg, it means they will give birth to a boy, XY. But if they give release X and X, they will give birth to a girl. So all these are still under chromosome. Now another point is allele. What is allele? Use the AL, alternative, then ELES. Just put GA as if you want to write Jill, but gene. That means alternative pairs of gene, alternative forms of gene. That's the meaning of allele. And these alternative genes, you have known the meaning of gene, the one that is carrying our behavior. So that gene carrying our behavior, it has alternative like pairs, the one that it can pair with. So, and these alternative gene, they are on the same locus, just like twins that are on the same, inside the same womb. So at least they are alternative gene that are on the same locus. Use the letter L to remember locus, on the same, and that same you can use letter S. So on the homologous chromosome. So, and which pair during meiosis? Like I said, like twins, they pair. So during meiosis, that's a kind of reproduction process. So during cell division, so you see that there will be a change. They will begin to exchange. So that means, for example, assuming this chromosome here, so this and this, they begin to exchange, they work together. Now another term is genotype, from the word gene. Genotype, meaning that the genetic constitution is called genotype or genetic makeup. It means what makes up you as a person is your genotype. All the things you got from your parents will make up you. That's your genotype. So we can also call it genetic makeup. So that's genotype, and there are different types of genotype. We have AA, AS, SS. AA, someone that is not having sickle cell, but AS is a carrier of sickle cell, though he's not having the sickle cell because he's the dominant one. I mean the dominant one is A. The S is just like a carrier, and that's why you can call this person a carrier of sickle cell, but he's not actually having the sickle cell. While this one is the person that is really having the sickle cell, so that is the SS. So talking about sickle cell is the major thing that always affects blood. So it affects blood, and that's why this kind of person falls sick easily. So that's a sickle cell person. So and also in looking at this particular genotype SS, it's always been advised that person that has this SS should not marry another person that has SS, because they will give birth to a sickler as their product. Then the same thing, AS is not advisable to marry AS because SS can also still be produced, but AA can marry any of these two. AA can marry AS, AA can marry SS. So that's for the genotype, which is the genetic makeup. Then another one is phenotype, from the word physical, PH, physical. It means the physical expression of genotype is phenotype, meaning that the genotype that someone has can be expressed through phenotype. Maybe for example, if the genotype is SS, phenotypically it will appear in the appearance, it will manifest when it comes to manifestation of sickness and the likes. So then also if one's genotype there is a kind of tallness in the gene of someone, that's part of the genotype, then that tallness will manifest physically. So meaning that phenotype is the outward expression of the genes, so and it's determined by the interaction of its genetic constitution and its environment. It means it relates to its environment. So once again, phenotype, physical expression of the genotype. Then there's another thing called linked gene, from our chromosome where we have gene. So every gene that is together with another gene is called linked gene. We find it on a chromosome. Then there's another one called sex-linked gene. When we hear sex-linked gene, it means those ones that are situated on the X of the chromosome, because looking at chromosome, we say we have XY for a male. So that X, once there is a kind of character on this X that can be inherited, that character that can be inherited, it will be on the X. So for a man, so it won't affect the Y. Then for a woman, XX, it means it can be on any of the two X's. So that's a sex-linked gene. And what are those sex? There are some characters, we call them sex-linked characters. They stay on this X. So for example of this sex-linked character, we have the color blindness. We also have the hemophilia. So this color blindness, if you want to know if someone is color blind, that's in terms of when the person sees red, the red will look like green. When he sees green, it will look like red. That's color blind. He will not be able to differentiate between green and red. So if someone is color blind, you will see letter N, small letter N on the X. So but if the person is normal, that is not having, it will be capital letter N. So for example now, for a man that is color blind, you will see XY, but on top of the X there will be small letter N for a man. So this person is color blind. But if the person is normal, for a man it will be capital letter N. While for a woman, so you will see the same 2X here. So if the woman is color blind, you will see small n on both sides, small n and small n on both sides. That's for a woman that is color blind. But if the woman is normal, it means you will see capital letter N on both sides for a normal person that is not having color blindness. And there's another one called carrier. It means if there is no n, small n, and there's also big N, meaning that this person is a carrier of color blindness. That means it has a sign of color blindness, which is small letter N, while the other one is the normal one. So that's for sex-linked genes. Then the next one is pure line. When you hear pure from the word, just like pure water that doesn't have impurity, doesn't have dirty thing in its pure line. So meaning that for this pure line, you will have homozygous. Homo means the same, just like this small letter y, both of them small letter, that they are the same, homo. So it means if there is homozygous, either they are recessive, like this one now is recessive, while this one is dominant. Dominant is the one that is always manifesting at a particular time, but recessive, it will be hiding like mask, but will later be revealed in future. So when you have these two set of things crossing, then that's a pure line. So for example, if I say you are crossing big y with small y, that's a pure line. But if it is capital letter and small letter, it is not pure. For example, assuming this one is big Y and this one is small y, crossing it with another big Y, small y, that one is not pure. So they must be the same, maybe the same capital letter, the same small letter. That's a pure line. Just like two professors from one university with another two doctors from another university, so trying to have a kind of competition, that's pure line. But if it is a professor and a doctor from one university, a professor and a doctor from another university, that is not a pure line. So it means they must all be homozygous in pure line. So either they are dominant or they are yes, dominant or recessive. Then another word we have is homozygous. We have said homo is the same, homozygous, while hetero is different. It means when the alleles are identical, that's homozygous, like this big T, just like that pure line, they are homozygous. But when they are different, dissimilar, that's heterozygous, like big T, small t, that one is hetero, they are different. Now another term we have is immunity. So that's the ability of an organism to combat infection by parasites. So that's ability to combat. So it means that for every human being there's an immune system. What normally fights disease or stranger or those pathogens that may come into the body. So ability to fight those strangers are called immunity. Then the next one, we have mutation. Mutation is a sudden change, maybe the gene in the human body, so suddenly change due to some reactions, or maybe relation with the environment, so or maybe in the process of meiosis, so sudden change that happen to the gene or the DNA. So that's mutation. And in this sudden change, maybe in a particular generation, they are always producing tall persons, but because of the sudden change, so it means it may give them shortness. So it means sudden change in the DNA or gene. Then we also have antigens and antibodies. This antigen is just like ID card on the body of a thief, antigen, while antibodies is now like a security man of a particular house. So it means that when the security man, which is the antibodies, when this security man antibodies sees the antigen, that means the ID card of a thief, then there will be a stimulation. It means this antigen always stimulates production of antibodies. That means the security guard, which is the antibody, will quickly have to come and raise, or to come and fight the thief. Who is the thief? The thief represents a pathogen. When a pathogen comes into a body, it causes disease. Pathogen will cause disease. So it is on this pathogen that antigen is always hanging. Like I said, that's ID card. Antigen is like ID card that is on a thief. So when the thief is having the ID card, so which is the antigen as the ID card, the pathogen as a thief. So when this antibodies, which is the security man, sees the ID card antigen, it will know that, oh, this is a thief, then it will attack it. So meaning the antibody always attack the antigen. It attacks the antigens. So and once the antigen has been attacked, that means just like the security man will collect the person's ID card, that's the attacking of the antigen. So we'll collect the ID card, and once the ID card is collected, then the thief has also been apprehended. Now we have then antibodies, like I said, that's the security man. So it's always in form of protein. So antibodies in our body is in form of protein. And take note, in our body, these antibodies which I call security man. So there is another name they give to them. So for example, this antigen, you can say when there is a kind of blood, a wrong combination of blood, we have what we call agglutination. So meaning that we can call antigen agglutinogen from letter G. Why antibody? We call it agglutinin. So these antibodies is from the word agglutinin, is a protein. Is a protein production. Is always responding to the presence of foreign substance. So it's always a foreign substance, and that's antigen. When it sees the antigen as ID card is coming with a pathogen, so it will fight it, just like anti-robbery squad that always fights the robbers. So that's the work of antibodies. Then another one is dominant gene. When you hear dominant gene, it's one of the pair of contrasted characters which is fully developed, whether the individual is homozygous. So the major, when you hear the word fully developed, the key term, you can just use this direct definition that dominant, something that is expressed, character that is expressed, that's a dominant. So any character that is expressed is a dominant character. So that's for dominant gene. Then it's just like maybe there are two sets of people in a class, the boys and the girls. So when the boys are making noise, it will overshadow that of the girls. That's what we call dominant gene. So but when the boys keep quiet and the girls are now making noise, it means the girls own were what, recessive in the first place, but later it become dominant. So what does that mean? For example is maybe when a family, they give birth to someone that is dark. So maybe the man is dark, the woman is also but the woman is albino. Let's say the woman is albino, the man is dark. So when they give birth, they now give birth to a dark child. So that dark child now marry another dark person. So and they now give birth to albino. So the albino they gave birth to, it's not as if that child is a bastard, or but what happened is that it was the grandparents that one of them was albino. So just that the albino was recessive, it did not manifest. It was the dark of the darkness of the, or let me say the dark color of the father that manifested in the child, but the albinism is still recessive in that child. But in the future, albinism now manifests. So meaning that there's what we call dominance. What manifested here was the dark color, but recessive, that means the albinism was recessive, it was masked, like masquerade, it was masked, but in future when that person now married, it now manifested. So that is what we call recessive. The one that was hidden and later manifested is the recessive. So but anyone that has always been expressed is dominant. So there is another term we use here, so which is recessive gene. So I've actually mentioned it. For dominant, if you want to know something dominant, it will be in capital letter, but anything recessive, it will be in small letter. So recessive gene is the contrasted characters, so which is developed in heterozygous individual. So if you want to know recessive, something is always in a smaller form, so you just use the word mask. The definition of recessive gene, this is the gene that is masked but will later be seen in the next or in the future generation. So recessive gene is masked, but dominant gene is expressed. So it's expressed or is seen. And I said small letter for recessive, capital letter for dominance. Now there's another thing called zygote. Zygote, that's the offspring after cells, male and female cells have come together. So the offspring, that's the baby, that's what we call zygote. Then gametes. Gametes is the male sex cell and the female sex cell. Talking about the sperm and the egg, they are the gametes. They are the ones that come together to produce this zygote. Then fertilization is now the process of how those gametes will come together. So this is autosome. So I've talked about autosome earlier, that autosome is the 22 pairs of chromosome out of the 23 pairs, so that will determine your eye, the type of eye you have, the eye, ear, the nose, the length of the leg, the color, every characteristics of human. So 22 pairs of chromosome carries that, but the only one pair, that one is for the sex chromosome. The only one pair will only determine if you will be a boy or a girl. Then locus means location. That's a site where your chromosome will stay, where it will occupy by a gene. So these are some of the terms we use in genetics. Now there's something we call transmission and expression of characters in organisms. So some characters are being transmitted. Another name for character is trait. Every human being has a trait, a character, or many characters, not just a character. The blood group is a character. That means maybe you are blood group A, blood group B, blood group O, or blood group AB. So all these are what characters in human. Apart from that, we also have the heights. So every human being has a particular height. That's a character. They have weight, they have skin color. So some people can roll their tongue. So some can taste PTC. So all these are characters in human beings. So there are still many characters. So and these characters, they can be transmitted. That means they move from parent to the offspring, and they can be expressed. So what are the there's something we call variation, which are the differences. What normally cause variation? Once there is meiosis, there will be exchange. That means meiosis, during maybe the male and female sperm and egg coming together, there will be an exchange of their genetic material. So when these genes are being exchanged, they will combine. There will be a kind of segregation, there will be a kind of independent assortment. So before you know it, you start seeing variation. That's why you see a child produced by parents is having some slight differences. So that's as a result of meiosis. There's also called crossing over from that meiosis. So it means from each chromosome, from one point to another, there will be a crossing over. So then another thing we consider in this particular topic is fertilization. When you hear fertilization, I've said it earlier that fusion of the male and the female gamete to form zygotes. So they come together, that's fertilization. Then transmissible character. That means those characters that can be transmitted. There are some characters that can be transmitted, there are some they cannot be transmitted. Any character that is carried in the gene, they can be transmitted. For example, when you look at hemophilia, hemophilia is something that goes along with gene, meaning that if one's parent has hemophilia, that child can also have it. So it means that character is transmitted. Then the same thing, color blindness is transmissible. So all these are transmitted. Also like diabetes, hepatitis, so all these things are characters that can be transmitted because they are carried in the genes. And it depends on which of the parents is carrying that dominant genotype. That means that particular character that is showing. Any parent that is carrying will determine where if it will be transmittable. So that means if it is dominant in the father or the mother, it will be transmittable. But if it is a recessive, it will not be transmissible yet, but it will still manifest. So these are all transmissible characters. So then other things that can be transmitted, sickle cell anemia, that's SS genotype, can be transmitted. Hemophilia can be transmitted. Colorblind, when you hear hemophilia, that means when there is excessive bleeding, that any minor cuts, there will be excessive bleeding. So and those ones that are not transmissible, that means that cannot be inherited from one's parent, that one is anything that has to do with environment. For example, maybe there is a loss of one leg in the course of an accident, that it means by the time that person gives birth, that does not mean the child also will not have leg. So all these are non-transmissible. Looking at Mendel's experiments, Mendel, that's Gregor Mendel, that's the father of genetics, is an Austrian monk. He used what we call garden pea, that's Pisum sativum, to perform an experiment on genetics. Why? Because he noticed that this Pisum sativum, the garden pea, it has several characteristics that can explain genetics, such as the height of the stem. So it has both tall and short. It has flower that is red and white, pod that is green and yellow. It has surface pod that is smooth and constricted. It has surface of testa that is round and wrinkled. It has position of flowers that is axil and terminal. It has color of seed that is yellow and green. So because of these several characteristics that Pisum sativum is having. So he saw it as a good instrument that he can use to explain genetics, that's in terms of plants. So then there's another thing we call breeding through. When you hear the word breeding through, once a stalk of red flowered pea plants that always produce red flower offspring. So when you cross it among themselves, that means you cross red flower with red flower. So it means and you have breed through. That's you bred through. So crossing red with red, that's you are breeding through. So it has two genes for red color, big R, big R. That's the dominant are red. So this red flower plant form a pure stalk. From the word pure, that means no dirty things. That's meaning of pure, no combination. So it's just the same thing that is produced. The same red you planted is the same red that is produced. So that's a pure breed. So then a stalk of white flower pea plant that always breeds through has two genes for white color, small r small. It means this one too is also a breeding through or pure line. Once they are the same, that's breed through or pure line. If you plant white, you see white. You plant red, you see red. That's breed through. Now, when you now want to cross this red and white, so that is monohybrid cross. Mono means one, so that means that red and white still represent only color. But if you are crossing something that has to do with both color and shape, maybe you say this one is wrinkle, that one is round, this one is red, that one is dye. Hybrid, dye is two. So monohybrid is just crossing one factor. For example, here now we are using that same color that we have here, only color. So and the color we are using is red and the other one is white. So let's see how the monohybrid cross takes place. So this is the parents. That means the first one that representing the father, that's red for the red plants. We are using plant now for flower. Then the other thing that we are crossing it is white. It's white, but don't forget they are both homozygous. Homo is the same, big R, small r, small r. So this is a kind of pure breeding, true breeding, because they are both pure. So from the father, remember, just like sperm, so one will be released, big R, which is the red. Then one will also be released, small r, which is the white. So by the time they cross, they will give you everything will be red. But what kind of red is that? They call it heterozygous red, meaning that there is a combination where big R will join with small r. That's big R, small r. The same big R will still cross with the second small r. That's big R, small r. Then the second big R here will cross with small r, big R, small r. And finally that big R again we cross it with the last small r, that's big R, small r. You can see that all the four, they are big R, small r, big R, small r, big R, small r. So but we still call all of them red because the big R is dominant over the small r. Small r is recessive, that means it's not showing, but the one that is showing is the big R, and that's why we call them all red. The four of them, they are red, or you say they are 100% red. So 100% red, or you say they are all red. Four of them are red. Now all these children or all these products, it means all the product of that crossing of red and white, the four of them, so we call them F1 generation. That means the first generation, just like the first set of fowl, chicken that we're giving birth to, the chicks produced by the cock and the hen. So the first four children they gave birth to, so we call them F1, first generation. So looking at this, it means we are after only color. It was color that was produced as a monohybrid crossing. Then we can progress to to the second generation. So it's still on monohybrid. It's still on monohybrid. We have seen the first generation from the monohybrid. But if you move to the second generation, that's when you now take the product of the first generation, big R, small r, with another big R, small r. By the time you cross them together, we have different changes coming up. So take note, this big R, small r is called hetero. This is an heterozygous. So these two heterozygous, when they cross, it means big R, small r, then big R, small r here, small r, small r. So these two, four products. Now there are two different things we can get here: genotypic ratio from genotype. Then we also have phenotypic ratio, phenotype. So the phenotypic is the one you can see from the word physical. So I can see dominant red here 1, dominant red here 2, dominant here red 3, meaning that that's 3 ratio. This one does not have dominant. So we call it 3:1. That's for the phenotypic ratio. That means physically we can see 3 red so and 1 white. So that's 3:1. Then for the second one, which is the genotypic ratio. So this one is the same thing we can see. This one that is the same is only 1. This one that is also the same here, they are 2. That means big R, small r, big R, small r, they are 2. Why this one is small r, the only different one 1. So it means that the genotypic ratio is 1:2:1. So these are the product of the F2 generation. That's crossing, doing your monohybrid cross. And take note, this kind of question is common in WAEC and JAM, that they always ask that when there is a crossing, that's maybe a pure breeding, a true breeding, a pure line breeding, that what will be the numbers of the products, that if they have 4 children, what would they look like? So take note, is all that which is dominant, assuming they say crossing between tallness, let's say big T, small t, and shortness. Maybe the man is tall, the woman is short, that when they give birth to 4 children, so if the tallness of the man is dominant and the shortness of the woman is recessive, that if they give birth to 4 children, how many of them will be tall? You say 4 of them will be tall, because this big T will be dominant over the small t. So then they can also ask in terms of percentage. You say 100% will be tall. Then if they say what of the second generation, so you start bringing it from here, that maybe the one that will be red, you say 1/4 will be red, or 25% will be red, which is homogeneous red. But they say what of heterozygous red, you say 2 of them will be red, or heterozygous red, that's 50%. Then if they say what of white, that's 1/4 or 25%. So these are the way they always ask questions in WAEC. So then another thing, as we have seen from here, so we have seen the crossing, how they cross in the monohybrid crossing. So all the answers as the result of my explanations, they are here. The phenotypic 3:1, the genotypic that's 1:2:1. Then there's another thing we call test cross or back cross. What is test cross or back cross? That means when you take this is done to find out the phenotype. This one, the main reason why we are doing back cross is like reversing, is that you want to find out the phenotype of each red flower pea plant in the F2 generation. So trying to get the phenotype of each red flowered pea plant in the F2 generation. So this is carried out by crossing the red flowered F2 phenotype, so with a true breeding white flowered plants genotype from the P generation. For example, look at the parent plant of the big R, we have crossed it with the small r, so the product, we said they are all heterozygous red, which are 4. So this one here can be used for back cross. How will it be used? That means you pick the big R and the small r that is here, and then join it with the small r and small r from the beginning. Remember, from the beginning it was only big R and big R, small r, but here if you want to do back cross, pick from the children and then pick from the parents. So the parents has small r, the children has big R, small r. So when you join these two together, you can use it for the back cross, so that you can use it to find out phenotype. You want to use it to find out the phenotype of each of them. So it means when you pick big R, small r, crossing it with small r, you want to do your back cross. You can also do the same thing like this for big R and big R, small r, to use it for back cross. That means to find out the phenotype. Now here we can see it, that we have taken the big R, small r, that is one of the children, crossing it with one of the parents. So and when we did that, big R joined with small r, big R joins with small r, then small r with small r, another small r with small r. So this one we call it red, that's heterozygous red. Then this ones, they are white, homozygous white. So meaning that we have gotten back our red and our white. So that shows that we have gone back to the phenotype. So that means we have seen that it was 2 reds and 2 white. So we started with the same red and white. See, this is 2 red and 2 white, so it means we have back cross to get back our red and white. The same thing, which is still 1:1. So the same thing can be done to the other one. I underline 2 things from the beginning. So when you do that, you also get this red and white from the crossing. Now the next one, another point we want to look at is what we call incomplete dominance. So when you hear incomplete dominance, so in incomplete, it means one is not completely dominant. For example, or you can use the word blending inheritance. So blending inheritance. So that's incomplete dominance. It's different from codominance. Though what they wrote here is not the correct thing. So it's a blending inheritance. Maybe, for example, there's a fowl. They call it Andalusian fowl. So when you take the white of the fowl, which is homozygous, with a black of the fowl, which is homozygous, by the time they cross, it is blue. You will be seeing. That means the W and the B coming together, that's BW. So instead of saying white and black, it's blue. So the blue means it has changed the color of the black, it has changed the color of the white. So it means they are blended together. So meaning that that's incomplete dominance. They will blend. You will not see the specific color. So they would have merged together. So that is incomplete dominance. The same thing, another plant, apart from this Andalusian fowl, for plants, we have 4 o'clock plants. So from the 4 o'clock plants, we use red flower and white flower. So when they cross, it means you won't see red, you won't see white, but you can see pink. It means the red has blended with the white to give us pink. So from the red, we have big R, which is homozygous red, and we also have white, big W crossing. So they will give us pink. So that is an example of what we call incomplete dominance. Then another one here, we can see the workings of that explanation. Now codominance, which is different from incomplete dominance. Codominance is commonly used for blood group like ABO, meaning that blood group A, blood group B, blood group AB, blood group O. So this blood group now, that's ABO blood group. In man, it can exhibit codominance. What's the meaning of co? Like fellow dominance. That means A will dominate his own, B will dominate his own. So both of them, each of them will dominate. Someone that has blood group A, if you transfer or transfuse the blood of that person into someone that has blood group B, the A will try to fight, B will also try to fight. That means both of them will become dominant in that person's body, and they will start causing agglutination and can cause death. So that's codominance. So now the blood group is determined by a single pair of genes, single pair located on a homologous pair of chromosome. There are 3 different alleles in this codominance. So what are the 3 alleles we have? We have the BTA, so for antigen A. Remember, we have said antigen is just like the ID card on a thief, which we call pathogen that can affect someone's body. Then we also have the TB. So the TB, which is also for antigen of B. Then we also have the small TO, which is not having any antigen. Meaning that anybody that has blood group O, there is no antigen in the person's blood. Then anyone that has blood group B, it has antigen. Anyone that has blood group A, it has antigen. So there are different antigens. So there's an antigen for A, antigen for B, but there's no antigen for O. Now let's see, take notes for O is always recessive. So let's see the blood groups and the antigens or the allele. Now someone that has blood group A, we can see, or let me just use a particular thing to explain all those ones from this particular page here, so that we will not repeat all this. So for the antigens that we have here, so we have, there is a particular explanation under this that can explain all these antigens and then the antibodies. So they are explained side by side, so to give a better explanation. Now as you can see from this drawing here, so everything is listed here. So the antigen and the antibody. So from here, anybody that has blood group A, it means there is an antigen A on that person's body. For example, maybe a man called John and another man called Peter. So this John, his blood group is A. So the blood group of John is A. So this John now has an antigen A. That means ID card of A. So meaning that that ID card is on this John, seeing this John as maybe a thief. So meaning that the antigen A, which is the ID card on this John representing the thief, there will be an antibody. It means this antigen A is actually in the person's blood. So then the antibody is in the white blood cell, or that's the plasma. So the plasma of this person has antibody, but the antigen is in the red blood cell. Okay, now this person we call John, we said he has this antigen A in his red blood cell, which is representing ID card on a thief. Then the antibody of another person, which is now the antibody of another person. So let's say the person is having anti-B. So meaning that this B will fight the A, because the antigen A has stimulated this antibody to fight the A. So meaning that anywhere A is found, B will fight it. So antigen is A, just like it is always steering up an opposition. So you can see say antigen A like a rat, then antibody B like a cat. It means whenever a cat sees a rat, it will show up his anger. So meaning that anywhere you see antigen A, the antibody B will fight against it. So still looking at blood group A. So the possible genotype of this blood group A is IAA. That's dominant for the A. So or A and O. Why? It means anybody that has blood group A has this A and A, and then it may also have O. Because the O has no antigen. That means it's this A can relate with A, and this A can relate with O. So someone that has blood group A, so you can transfuse blood group A to the person's body. The same way, someone that has blood group A, you can transfuse blood group O to the person's body. So that's for possible genotype. Then this person that has blood group A, so it is a donor. So his donors could be A or O, like from what I said here. That means donors, those that will give blood to the person that has blood group A. A can give to him, O can also give to that person. Then recipients. So the recipients are A and AB. That means this person that has blood group A can give someone that has A, and it can give someone that has AB. Why? Because this one that has AB, when it gives A to him, it can merge with the A in the AB. So it means this blood group A can give to A and AB, while this blood group A can receive from A and from O. It means they are donors. The donors that will donate to blood group A, they are A and O. So and that's how it applies to B also. Anybody that has blood group B, it means the antigen of that person is B. That means the ID card that will steer up the enemy of A. So it means if the person has B, A will be the enemy, so which is the antibody. So then the possible genotype, they are BBBO, meaning that it can receive from B. This person that has blood group B can receive from B. The person from blood group B can also receive from O. And that's why their donors are B and O, those that will donate to him. But the one he will not donate to, that will give out to, so someone that has blood group B can give it to B, can give it to AB. Then someone that has blood group AB, so that person has antigens A and B. Antigens A and B. So since he has antigens A and B, he will not have any enemies. He will not have enemy like antibody, because if he's having antibody of A is already here. The A and A cannot fight. If he's having antibody of B, this is B here already, so B and B cannot fight. So meaning that there is no antibody for these antigens A and B. The possible genotype is also still A and B. So meaning that this person that has A and B, he can receive from A, he can receive from B. So meaning that those donors, those that will donate to AB, they are A can donate to it, B can donate to it, AB also, when you join them together, they can also donate to this person. Then O can still donate to this person. So that is that the recipient. Who can this AB now donate to? AB can only donate to AB. Then the last blood group we have here is blood group O. So this blood group O individual, so it's antigen, it has no antigen, that's nil. Then the antibodies, so that means those things that can fight this blood group O is A can fight it, B can fight this person. So it means he has no antigen. So A can fight it, that means there is nothing he can relate to it. B, he doesn't have any B to relate to, so they can fight this person. So meaning that anybody that has blood group O, you cannot give the person A, you cannot give the person B, otherwise it will fight them. Then possible genotype, it means it can only receive from only the person that has blood group O. But and in terms of donors, so that means those that will donate to blood group O, there are also people that have blood group O. Then recipient, those that will receive from blood group O, A can receive from O, B can receive from O, AB can receive from O, O can also receive from O. So the antigens are both, we call them agglutinogens, those antigen. Then the antibodies, we call them agglutinin. So antigen would not have similarly named antibody. So that's, they will not have similarly named antibody. That means if the antigen is A, the antibody will be B. So they will not have something similar. And take note, when there is a wrong connection, if A and B are coming together, there will be agglutination. That's a mismatch and can cause death. So then take note of this particular there are 3 points I want to mention before we round off this aspect. Like when we start our crossing, there are some crossing that will carry these symbols, there are some crossing that will carry these symbols. So these ones that carry these symbols, for example, we have here the albinism. Like when someone is having capital letter A and capital letter A, we call it normal. That means the person is not an albino. Then someone that is having big A, small a is a carrier. Someone that is having small a is an albino. So these are, when we start doing the crossing, take note of that. Then for the sickle cell, when someone is having AS is a carrier, AS is a carrier, but AA is normal. So why SS, someone that is sickler. So take note of that. Then the last one is color blind. So as I've said, if you see small letter N on the X, the person is color blind. But if you see capital letter N, but if it is combined together like this, means this person is a carrier. The color blind is here, normal is here. But if both of them are small n, then that's color blind. So that is how it is. And take note, it is commonly affecting male. It is the men that usually have that color blindness. So the next class will be continuing with this study. So we the points where we are going to continue, so that will be from the dihybrid crossing. So we have seen the monohybrid crossing. So the next class we'll be looking at the dihybrid crossing, and we also look at the probability and the likes. So thanks for listening.